Hello students, myself Dr. Mohammad Zafar Iqbal. I am from Government Arts and Science College, Karwar. In this video, we are going to discuss about the genetic code. Genetic code is one of the most important topic in genetics as well as in molecular biology. And this is very important to understand the protein biosynthesis. That's why if you want to understand the protein biosynthesis, then the first step is to understand the genetic code. That's why this particular topic is very important. And in this video, we are going to discuss about the some of the very important concepts related to the genetic code. And before we get into this particular topic, let us set some of the objectives for this particular video. And in this video, we are going to understand the central dogma of molecular biology. What exactly the central dogma of molecular biology says, that we have to understand. This is very, very important. And if you take complete molecular biology, the complete molecular biology revolves around this particular concept, that is the central dogma of molecular. That's why the central dogma of molecular biology becomes very, very important. And second one is that we are going to understand what is the meaning of or the concept of the genetic code. We are going to understand what exactly we mean by genetic code, that we are going to understand in this particular video. And third, we are going to understand different features of the genetic code. If you take the genetic code has got certain universal features. And you take any organism, in all organisms you will find these features. That's why they are called as the universal features. That's why we are going to discuss all those features in this particular video. And in this video, we are also going to discuss about the very, very important concept called as the degeneracy of the codon. And the degeneracy of codon is very, very important. And because of this, we are able to have, that is the message for the all the amino acid, that is the 20 amino acids are there, and we have got only 4 nucleotides are there. And in order to give meaning for each and every amino acid, we need to have a more number of codons as compared to the that is what we call them as a amino acid. That's what this particular thing is called, the degeneracy of codon. That also we are going to understand in this particular video. And at the end of this particular video, we are going to understand what is the meaning of the oval hypothesis. These are the some of the important things which we are going to discuss in this video. First, we discuss about the central dogma of molecular biology. As I already said, that if you take the molecular biology or you take the genetics, the whole genetics or molecular biology revolves around this particular concept of the that is the what we call it is a central dogma of molecular biology. And the central dogma of molecular biology always starts with the DNA. Here you can see that this is the DNA. DNA is the central molecule, and we know that DNA is very, very important because this is the molecule or the biomolecule which carries the genetic information. Since this is the molecule which carries the genetic information, that's why the integrity of the DNA is very, very important. It should be integrated and should not lose the meaning in the during the process of the replication. That's why the integrity of the DNA molecule is very important. And if you take the central dogma of molecular biology, the central dogma of molecular biology starts with the DNA. And now, if you take the DNA, DNA has got two fates. So the one of the fate of the DNA is to undergo replication, because we know that since it has to be passed to the next generation, and that's why in order to do that, we can find that DNA has to be replicated. That precise replication of the DNA is very, very important, and there should not be any mistake. And that's why whatever this particular replication process there, it must be foolproof. And that's why this is one of the fate of the DNA molecule. Then the second fate of the DNA molecule is to get that is the transcribed into the RNA molecule. Whatever the message which is present in the form of DNA, and we know that there are 4-letter language in the DNA, and these 4 letters are adenine, guanine, thymine, and the cytosine. And these 4-letter language of DNA must be converted into the language of the RNA. Again, the RNA has got 4 letters. One is the adenine, guanine, cytosine. Instead of thymine, we have got a uracil in case of the RNA molecule. That's why the second fate of the DNA molecule is to convert the language of DNA into the language of the RNA. And this particular process is called the transcription. And we know that the transcription is very, very important. If you take a synthesis of any RNA, you take so many different types of the RNAs are there. We have got some major RNAs, and we have some minor RNAs. And if you take all these RNAs, they must get synthesized from the DNA. There are DNA specific RNA polymerases are there. These DNA specific RNA polymerases are responsible for converting the language of DNA into the RNA. That's why this particular process is called the transcription. Here you can come across one particular term called as the transcription. The transcription is nothing but is the conversion of the language of DNA into the RNA is called as the transcription. And this is the first step in the central dogma of molecular biology. Now you can see that this is the, now what we have done, we have discussed about the DNA molecule, and we have discussed about the different type of the faiths which are associated with the DNA molecule. We also discussed about the that is the how DNA gets transcribed into the RNA molecule. And in case of the normal conditions, you can find that this is the only possibility, that is the DNA can only be converted into RNA. But in majority of the organism, it is not at all possible to convert the that is the RNA into the DNA. But there are certain viruses, and these viruses are called the that is the retroviruses. And whatever these retroviruses are there, these retroviruses are known to have a one particular process, what we call it as a reverse transcription. Why we call it as a reverse transcription? Because it is the reverse process of the transcription. What we are doing in the transcription, the DNA is getting transcribed into the RNA, and in the reverse transcription, what is going to happen? The RNA is going to be converted into the DNA molecule. That's why this particular process, that is the reverse flow of information, is called the reverse transcription. And the reverse transcription is only possible in case of retroviruses. There is a very, very special group of viruses, RNA viruses, and only these viruses have the ability to undergo reverse transcription. And whatever the reverse transcription is there, the reverse transcription is only possible because of the presence of an enzyme called as the reverse transcriptase. There is a very, very important enzyme, and this particular enzyme is called the reverse transcriptase, and this enzyme is responsible for converting the language of the RNA into the DNA, because we know that the retrovirus have to replicate in the host cell, and in order to do so, what they have to do, they have to integrate their genome into the genome of the host cell. That's why the RNA cannot be integrated into the genome of the host cell. That's why the host cell. That's why the RNA gets converted into the complementary DNA, and once it gets converted into the complementary DNA, automatically you can find that it can get integrated into the DNA of the host cell. That's why you can find that this particular process is only in case of the such type of the viruses. Then what is the normal fate of the RNA in majority of the organisms? You take all the animals, you take all the plants, you take all the bacteria, and you take a majority of the viruses. In case of all these, you can find that whatever the RNA is there, RNA gets translated into the protein. And this particular process, that is the conversion of the language of RNA. How many letters are there? The language of RNA has got 4 letters. That is the A, G, and CU. And this particular language of RNA should get converted into the language of the protein. And we know that the protein has got a language of about 20 amino acids. There are 20 amino acids are there. That's why the language of RNA must give information for the formation of a protein which has got 20 different type of the amino acids. That's why this is also very, very important, because we know that at the end we are interested in the protein, because the proteins are the molecules which give the function. And if you take the DNA or you take the RNA, they have the ability to give the information for the protein, but they themselves are of no use actually. And that's why you find that whatever the proteins are there, you take any trait or you take any characteristic feature, all these characteristic features are completely dependent upon the protein. That's why the protein is very, very important. And this particular process of the conversion of the RNA into protein is called the translation. Translation means converting the RNA into the protein is called the translation. This is also very, very important. And this is the second step in case of the central dogma of molecular biology. That's why if you take the central dogma from the molecular biology, it starts with the protein, then the intermediate molecule is the RNA molecule, and whatever the RNA molecule is there, the RNA molecule gets converted into the protein. And this is the normal flow of information. And there are two very, very important type of exceptions. One is that already we discussed, that is the reverse transcription, that is the conversion of the language of RNA into the DNA, that is by means of the retrovirus. In case of the retroviruses, by means of the reverse transcriptase enzyme is one very, very important type of the exception. And second type of exception is that there are certain viruses, and in such type of the virus, you can find that the genome is RNA, but they don't have a reverse transcriptase enzyme. Then what they have to do? They have a RNA dependent RNA polymerase. Please remember, the RNA dependent RNA polymerase enzyme, and this enzyme is only present in those RNA viruses which do not have the reverse transcriptase enzyme. And why? Only in that particular group of viruses it is possible to synthesize the RNA molecule. Otherwise, if you take animals, you take the plants, you take the bacteria, or you take other viruses, it is not at all possible to synthesize the RNA. That's why you find that only in case of this particular viruses which do not have RNA viruses which do not have the reverse transcriptase enzyme, they have another enzyme, and that particular enzyme is called the that is the RNA dependent RNA polymerase, and that is responsible for the synthesis of the RNA. These are the two exceptions, and apart from these two exceptions, you can find that if you take the central dogma of molecular biology, in case of all the animals, it will always start with the DNA. This is the molecule which stores the genetic information. This either gets replicated. In case of all the organisms, it must get replicated into its own copies. Then the second fate is to get converted into the RNA. Then RNA gets converted into the protein. The process of that is the converting the DNA into RNA is called the transcription. The process of converting RNA into the protein is called the translation. These are the important terms which are there in case of this particular central dogma of molecular biology. I hope you have understood what exactly mean by the central molecular, that is the central dogma of molecular biology, and this is very, very important. And whatever the molecular biology is there, the complete molecular biology depends upon this particular concept, that is the central dogma of molecular biology. That's why I ask students to give maximum attention to this particular concept and try to understand what exactly mean by transcription, what exactly mean by reverse transcription, and what is the meaning of the translation. These are some of the very, very important terms which are very much asked in the examinations. Now we will move to the genetic code, because this is this particular video is actually meant for the to discuss about the genetic code, and that's why we are going to discuss about the genetic code. Now first we will see the definition of the genetic code. The genetic code is a relationship between sequences of the bases in DNA or its RNA transcript and the sequence of the amino acid. And already said that whatever the message which is there in case of the DNA gets transcribed into the message, that is the RNA, and whatever the RNA transcripts are there, the RNA transcripts get converted, that is, they give message for the formation of the amino acid. And whatever this particular, how exactly that particular information which is there in the form of 4 nucleotides gets translated into the protein, that is called the genetic code. The genetic code is nothing but the set of nucleotides will give message for a particular type of the amino acid, and that particular relationship is relationship between the sequence of the base of the DNA and its RNA transcript and the sequence of amino acid is called as the genetic code. This is a very, very simple definition. I think there is nothing to worry about, and that's why try to understand, then it becomes easy for you to need not have to memorize, just understand what exactly it means, then automatically can reproduce in your own language. And you know that there are 20 amino acids are there. If you take standard amino acids, in all the organisms will find that there are 20 amino acids are there. There are some exception. One is the selenocysteine, second one is the pyrrolysine. That we are not going to discuss here, because the pyrrolysine and the selenocysteine are only present in the that is the in case of the methanogens, and apart from that you don't get in case of animals, in case of the plants, in case of the bacteria, in case of the viruses, we do not get the this particular these two type of the amino acids. That's why in majority of the organisms you can find that the proteins are made up of only 20 different types of the amino acid. Now if you have a 20 amino acid, then you should have a message for the formation of the 20 amino acid independently. And that's why if you take the nucleotides in groups of 2, that is there are 4 nucleotides are there, and if you take them in groups of 2, that is the 2 nucleotides as one codon, it will only give message for the formation of 16 different combinations. Only 16 different combinations are possible. More than this particular combination is not at all possible. That's why I can find that you can't take 4 nucleotides in groups of 2, then it is of no use, because it will give less than 20 amino acids. A message for the 20 amino, that's it, that is of no use actually. And in addition to the 20 amino acids, we should also have a signal sequences, that is the where exactly to start the that is the protein synthesis, and where exactly to stop the protein synthesis. All those signal sequences should also be there. That's why whatever the grouping up of 2 nucleotides as a codon is of no use, because it will only give maximum of 16 combinations. It means that it will only give message for the formation of a 16 different type of the amino acid, nothing more than that. That's why we have to move to the next step. Now we take 4 nucleotides and groups of 3, that is 3 nucleotides will give message for one particular amino acid, and there are 4 nucleotides are there. When you take 4 nucleotides in groups of 3, then automatically you can find that there are 64 combinations are there. It means that after 4 raised to 2, only next possibility is the 4 raised to 3. That is the only we can make a group of 2, or we can go for a group of 3. And when you take a group of 3, then automatically you can find that we have got a 64 combination. And whatever this particular thing is, it is much more than the required quantity, that is the 20 amino acids are there, but there are message for as many as 64 codons are there. And that's when you find that this appears to be true, and later on you can find that this was hypothetically calculated in the earlier period. Later on you can find that the scientists have clearly proved that there are 64 codons, and out of 64 codons there are 61 sense codons are there. Sense codon means these are the codons which give direct message for the amino acid, and there are four that is the message, that is the signal sequences are there. One of the signal sequences is called the start codon because it will give message for the start of the protein biosynthesis. And second, there are three stop codons are there, and these are the signals which are responsible for the termination of the protein biosynthesis. Altogether, you can find that 64 codons will be there, and out of 64, there are 61 of them are called the sense codon, and 4 of them are called the nonsense codon. Nonsense means they don't. It doesn't mean that they are nonsense. They have a message either for the start of the protein synthesis, or they have a message for the stop of the protein. That's why they are called the nonsense, because they don't code for the protein. That is the actual meaning of the nonsense. Whereas the sense codon means they have a sense for a particular amino acid. That's why you can find that this is the important thing which is to be remembered. Here you can find that some of the important ones we need to discuss. Now we will discuss about the certain features are there, and you take any organism, you take any plant, you take any animal, you take any bacteria or unique virus. In case of all these animals, organisms, you will find that these features are universally present. That's why these are called the universal features of the genetic code that we are going to discuss in this particular slide. And the first universal code of genetic is it is composed of the nucleotides of that is the triplet nucleotide that we call them as a codon. A codon is nothing but it is a group of three. In the earlier slide, we already discussed that when you take three nucleotides in a group, then it will give message for one particular amino acid. That particular group of three nucleotides is called as the codon. The codon is nothing but it is a group of three nucleotides is called as the codon. Since there are three nucleotides are there, that's why we call it as a triplet. This is the first universal feature of the genetic code. The second feature of the genetic code is the universal. What exactly mean by universal genetic code, that we will discuss. Next, third important universal feature is the non-overlapping. Each and every we are going to discuss in detail. That's why here I don't want to spend much time, and we'll just go through the list. And in the next slide, we will discuss each of these particular character feature in detail. And the fourth universal feature is the non-punctuation or the gaps. There are no gaps or there is no punctuation in case of the genetic code. Then the genetic code is said to be degenerate. Degeneracy is exhibited by means of the genetic code, that we call it degenerate. And the genetic code is ordered. Okay, then the seventh is the genetic code has got a stop codon and it has got a start codon. There is a message for the start where exactly the protein biosynthesis has to be started, and there is a message where exactly the protein biosynthesis is to be stopped. That is also very, very important. Otherwise, you can find that unnecessarily it will keep on copying the message for the protein, and it is more amino acid that will not be of any use. That's why stop and start codon are also very important. These are the features, and next we will go get into the details of this particular that is the feature. For the first is called as the composed of the triplet nucleotide triplets. That is the each codon has got three nucleotides. Three group of three nucleotides is called the codon. That we already discussed that if you take this particular group of two, only 16 combinations are possible. That is not at all enough. It is insufficient to code for the 20 different amino acids. Then what we have to do? We have to take the group of four nucleotides. That is the AT and GC we have got. And whatever this particular 18 GC we have to take in a group of this, then we will give it is possible to have a 64 count. That's why whatever this particular thing is there, it is true. And whatever the codon is there, the codon is nothing but in case of all the organisms. You take any organism, in every organism we can find that the codon means it has got three nucleotides. Group of three nucleotides is called the codon. That's why it is one of the universal feature in case of the genetic code. Then this is the just a table of the genetic code. See how 64 codons. If you count all these codons, you will find that there are 64 codons are there. And whenever you take this particular 64 codons, out of that you can find that there's a one codon, and this particular codon AUG is called the start codon. That is the initiator codon. AUG will always, the methionine is the first amino acid that is going to be incorporated. That is the starting point of the protein biosynthesis. And there are three stop codons. That stop codon means that is the message for the termination of the protein biosynthesis. One is the UAA. UAA is the stop codon. Universally it is present. UA is the second stop codon, and UGA is the third stop codon. Altogether, you can find that out of 64 different possible combinations, we have got 3 codons for to stop the protein biosynthesis. They are called the stop codon, and we have got 1 codon to start the protein biosynthesis, that we call it as a initiator codon, or it is also called the start codon. The start codon will always be methionine. That has to be kept in mind. That is also very, very important. And in this we can find that now in this particular table you can see that some of the amino acids are coded by means of more than 1 codon. Take for example phenylalanine. Here you can find that there are 2 codons. UUC also gives a meaning for the phenylalanine, and UUC also gives meaning for the phenylalanine. And in this way, here if you come to this particular category, there are 4 codons, and all these four codons give message for the leucine only. That is the leucine. Now you take this UCC gives meaning for the leucine, UC gives meaning for the leucine, UACA CUA gives meaning for the leucine. In this you can find that this is also one of the very important. That's why whatever this particular 61 sense codons are there, in order to give meaning for that, you can find that one amino acid can be coded by more than one codons actually. For example, leucine, there are 6 codons are there. All these 6 codons, whatever with these 6 codons, you can find that all of them will give meaning for the leucine only. But only thing is that one amino acid will have only 1 codon. Take for example CUU. The CUU only gives message for the leucine, but the CUU can't give meaning for the another amino acid. That's why you can find that there is a specificity is there. One codon exclusively gives message for the one amino acid, but one amino acid can have a more than 1 codon. That is also very important. I don't understand that one codon give exclusive meaning to a single amino acid. But if you take the one codon can have more than one amino acid can have a more than 1 codon actually. That's why this is also very important. And if you just carefully go through this particular table, you will understand what exactly the table means and what exactly the genetic code means. That is also one of the very, very important point to remember at this particular point. Now we'll go to the universal codons. And whatever the universal codons which say that genetic code is universal, what does it mean? The genetic code universal means if you take one particular codon, UUU. Now we'll take the example, some of the examples we'll take, and we can't take more than few examples. That's why we are going to take some of the example. And one of the example, first we are going to start with the UUU. And if you take the UUU, the UUU gives message for the formation of, for the incorporation of the phenylalanine. You take any organism, you take the animals, this particular codon means it is for the phenylalanine. You take plants, it means that if this particular codon exists, then it is message for the incorporation of the phenylalanine, even in case of the plants also. And you take the bacteria, even in case of the bacteria also, you can find that if this particular codon comes across, then it is a meaning for the incorporation of the phenylalanine. That's why you find that whatever this particular phenylalanine is there, the phenylalanine will be coded by means of a UU. That this is called the universal codon. It means that one particular codon will give message only for the only one amino acid. That is also very, very important. This point needs to be remembered. We'll take another example, that is the UAA. Whenever you come across a UAA, it may be in animals, it may be in plants, it may be in bacteria, or it may be in the virus. It means that in case of all these organisms, it will always give message for the incorporation of the isoleucine. This is the another second example. And we'll take another 2 example. One is the UGC. You take the UGC. Whenever you come across a UGC, it means that UGC will always give message for the incorporation of the cysteine. Whenever you take a UGC in a plant, if there is a codon called the UGC in the plant, it will give message for the incorporation of the cytosine. If there is a codon called the UGC in the animal, then also even in case of the animals also, it will give message for the formation of the cysteine. And in the same way, you can find that you take the bacteria, you take the virus. In case of all these organisms, if the codon is UGC, then it will give message for the formation of the cysteine only. That is that's why the inverse genetic code is called the universal. We'll take another example, that is the GCG. Whenever you come across a GCG in any organism, irrespective of their status, you will always find that GCG will always give message for the incorporation of the alanine. This is also important. That's why the second important feature of the genetic code is that the genetic code is universal. It means that if you take a particular codon, irrespective of the group of organism, it will give message for the that is the same amino acid. That is also there's a imprint. Then we will go to the genetic code. The next feature is called the genetic code is non-overlapping. What does it mean? It means that if you take this particular 2 examples, I have made 2 examples. Here you can find that 1, 2, 3 refers to 3 nucleotides of a codon. 4, 5, 6 refers to the 3 nucleotides of the second codon. 789 refers to the 3 nucleotides of the third codon. Now, if you see this, this is the condition in case of the genetic code. Here you can find that it is non-overlapping. Non-overlapping means 1, 2, 3. After 3, the 3 is not going to overlap with the fourth one, next codon actually. Group of first, second and 3 is one codon. Group of 4, 5, 6 is the second codon. Group of 7, 8, 9 is the third codon. And this is the actual thing which is there in case of all the organisms. You take any organism, you will always get that the genetic code is non-overlapping. In this way, you will get the genetic code. You will never get the genetic code in this fashion. If you take this particular example, here you can see what is the difference between these two. Here you can say that 1, 2, 3 is the part of first codon. 3, 4, 5 is the part of the second codon. It means that if you take this particular 3 is overlapping with the first codon and it is also overlapping with the second codon. And in the same we can find that if you take the 3, 4, 5 second codon, 5, 6, 7 third codon. Here also you can find that the 5 is overlapping actually. 5 is part of the second codon, 5 also part of the third codon. And in this we can find that whatever this particular condition is there, this particular condition is called the overlapping. Whereas if you take this particular condition, this is called the non-overlapping. Whatever the genetic code is there, genetic code is always a non-overlapping. This is the correct condition. And you take all the organisms, irrespective whether they are plant, whether they are animals, whether they are bacteria, whether they are viruses. In case of all these organisms, you can find that only you will get this type of the condition. You will never get a other type of condition. And in the same way, we can find that if it is this particular overlapping, it is not at all true. You will find that in case of none of the organisms, this type of the genetic code will be there. And only some exceptions are there. Maybe some, if you may ask, what is the condition for the pyrrolysine and the selenocysteine? In case of these two, you will find that only in case of some methanogens. I don't want to confuse you. Just leave it. Only in case of those animals, organisms, you can find that methanogens. You can find that in order to give message for the selenocysteine and its pyrrolysine, some kind of overlapping will take place by means of the looping. That's an exception. And apart from that, you can find that in all the organisms, you can find that this particular feature is true. That is the genetic code is non-overlapping. That is the continuous 1, 2, 3 is the first codon without any overlapping. 4, 5, 6 is 4 is the next nucleotide. 4, 5, 6 is the second codon. 7, 8, 9 is the third codon. And this is the feature which is present in case of all the organism. That's why this is the next important universal feature in case of all the organisms. Then we will go to the genetic code doesn't have a gaps or punctuations. What does it mean? Gaps and punctuation means here you can see that the genetic code is read in a continuously. It is called the open reading frame, ORF. What we call it is a open reading frame. And now you take two example. One example here you can find that is a without any gaps or without any punctuation. Now you can find that 1, 2, 3 is the first codon. There is no gap. 4, 5, 6. The next nucleotide itself is the part of the second codon. 4, 5, 6 is the second codon. And now you take the 7. 7 is the next nucleotide after 6, and 7, 8, 9 is the third codon. Now you can find that in between these there is no gap at all. You don't find any gaps. You don't find any commas. You don't find any full points. You don't find any punctuations. And that's when you find out whatever the genetic code is there, the genetic code in case of all the organism is the non-overlapping, or don't have any gaps, or it doesn't have any punctuations. This is the condition. Now what is the meaning of the gap? Now here you take the example of just in order to understand. It is not there, just for our better understanding we are taking this particular example. Now you take this particular example. Here you can see that 1, 2, 3 is the 3 nucleotides which give message for the first amino acid. Now what is happening? There is no fourth one is skipped. It means that it is a gap, and this particular gap is there. And after this particular gap, you can find that 5, 6 will become the nucleotides which give message for the second amino acid. It means that this is the second codon. It means that whatever this particular fourth is there, the fourth is a gap, or it is a punctuation. This type of the condition will not be. That's why in none of the organisms you will get this particular condition. That's why it is not at all true. That's why I can find out whatever this particular no junctions or no punctuation is the condition which is there in case of all the organism, irrespective, it may be plant, animals, bacteria, or virus. You can find that the genetic code always has got it doesn't have a gaps, no punctuation. This is the also another important universal feature. Now we'll come to the degeneracy of the codon. What does it exactly it means? And already we have said that the degeneracy of the codon is nothing but it is the if you take we have got 64 codons, okay. And if you take 64 codons, out of 64 codons, there are 61 codons which give message for the incorporation of the amino acid actually. and it means that more than one codon is responsible for giving message for the one amino acid actually. Take for example, we already discussed about, we'll also continue in the next slide. Here what we have to understand is that the number of codons are more and the amino acids are less. That it means that more number of codons should give a message for the one particular amino acid. That we already discussed about alanine, we discussed about the valine, we discussed about the isoleucine. So many examples are there. If you take all those examples, you can find that there is a degeneracy is there. And in case of the degeneracy, you can find that the first two interactions between the codon and the anticodon of the TNA molecule will exhibit the Watson and Crick base pairing. They strictly follow the Watson and Crick base pairing, and whatever the third nucleotide of the codon and the first nucleotide of the anticodon, they don't show that particular type of the stringency. They are flexible, and because of that particular flexible, you can find that whenever you take the third codon, if there is a degeneracy there, the third codon will be different. That's why you can find that this is called the degeneracy of the codon. And there are two types of the degeneracies. One is called the partial degeneracy, and second one is called the complete degeneracy. What exactly mean by partial degeneracy and what exactly mean by complete degeneracy, that we are going to discuss in the next slide. Now we'll take the complete degeneracy. Now I have made some kind of manipulations here. You can find it just so that you can understand it in a very easy. Now you can find that I have made certain groups as the rounds actually. And what are these? You take the leucine. These are the four nucleotides, four amino acids, and four amino acids are coded by means of four different type of the that is the codons actually. Now if you observe the first and second, in case of all these four, C and T are common to all the four codons. But when you take the last this thing, you can find that there is a difference. If T is present, still it will give message for the leucine. If cytosine is there, then also it will give message for the leucine. If adenine is there, then also it will give message for the leucine. And if there is guanine is present, then also it will give message for the incorporation of the leucine. It means that the third is not so significant, but first and second are very strictly maintained, and the third one is slightly some kind of a flexibility is exhibited. And whatever this particular phenomena is there, this particular phenomena is called as the complete degeneracy. In case of the complete degeneracy, what will happen? You have whatever be the third codon, it may be a purine or it may be a pyrimidine. Then also it will give the message for the same amino acid. This is the called as the complete degeneracy. Whatever be the third nucleotide of the codon, still it will give a message for the same amino acid. That is called the complete degeneracy. You take the example of serine. You take the example of serine. Here you can find that TCT gives message for the serine. TCC gives message for the serine. TCA still gives message for the serine. TCG still gives message for the. Here you can find that whatever be the purine or pyrimidine at the third base, you will find that it will give still the message for the formation, that is the incorporation of the serine only. And in the same way, we have got prolines are there, we have got threonines are there, we have got alanines are there, we have got valines are there, we have got glycine, we have got arginine. If you take all these are the group of codons which exhibit the complete degeneracy. In case of the complete degeneracy, whatever be the third codon, whether it may be a purine or it may be a pyrimidine, still it will give message for the same amino acid. That is the meaning of the complete degeneracy. Then what is the meaning of partial degeneracy? If you take this is the partial degeneracy. Now what is happening? If you take these two, phenylalanine and leucine, the first two of the phenylalanine, that is the TTT, gives message for the it should be UUU. This I have taken from the, this is the DNA actually. UU, if you take the UU, it will give message for the phenylalanine. UUC, it will give message for the phenylalanine. But if you take the, what are these T and C? The T and C, both of them are pyrimidines. And when you chain a pyrimidine with the purine, then automatically can find it, it will give message for the some other amino acid. Take for example, you replace this thymine pyrimidine with the adenine, then it will give, instead of coding for the phenylalanine, it will give message for the leucine. And the same way, you take the cytosine and you replace this particular pyrimidine, cytosine pyrimidine, with the other purine, that is the guanine, then it will give message for the some other amino acid. It means that in case of the partial degeneracy, the partial degeneracy is only true for the, if there is a pyrimidine, one pyrimidine is replaced by means of a second pyrimidine, still it will give message for the same amino acid. But if you change pyrimidine with the purine or purine with the pyrimidine, then it will give the some other meaning. That is the partial degeneracy. That's why I have made groups. This is the one example of a partial degeneracy, that is the phenylalanine and leucine. Here you can find it two first and two second nucleotides of the codon are present in all the four codons, but only change is the last third codon actually. If one pyrimidine is replaced with the second pyrimidine, still it will give meaning for the same amino acid. But if you change one pyrimidine with the purine, either A, if you replace T with A, then it will give leucine, or if you replace T with the G, then it will give message for the leucine. In this way, you can find that this is called the partial degeneracy. This is one group of partial degeneracy. Here also you can find that tryptophan. That is the UAU, it will give message for the tryptophan. UAC gives message for the tryptophan. UAA, one now what is this? This is the pyrimidine. This is also pyrimidine. Now you replace this particular pyrimidine with another pyrimidine. Still it will give message for the same amino acid. But when you change this particular pyrimidine with the purine, that is you replace the T with the adenine, it will give message for the stop codon. And you replace this T with the G, then it will give message for the another stop codon. In this way, we can find that the message itself is changing. That's what is called the partial degeneracy. In this way, we have got 3 more examples, and if you carefully observe these examples, then you will come to know that what exactly mean by partial degeneracy. Now try to understand what exactly mean by the complete degeneracy and what is the meaning of the partial degeneracy. In case of the complete degeneracy, you replace any nucleotide in the third position, still it will give message for the same amino acid. That is called the complete degeneracy. You replace pyrimidine with the pyrimidine or pyrimidine with the purine, then it will give same message as for the same amino acid. But if it, in case of the partial degeneracy, you can find out if you change pyrimidine with the pyrimidine, then it will give same message. But if you change the pyrimidine with the purine or pyrimidine with purine with the pyrimidine, then you can find out it is going to completely change the meaning. That's why you can find out this type of degeneracy is called the partial degeneracy. Now, stop codon. Already said that the next important feature is that the genetic code always starts with a one particular codon, what we call it is a AUG, that is the methionine. The first codon is the methionine. If you take the first codon AUG, it means that this is the starting codon, the initiator codon, and AUG will always give meaning for the methionine. Whenever you get AUG, it will always give a message for the methionine only. And this particular codon is called as the start codon, or it is also called as the initiator codon. Start codon means this is the place from where the protein biosynthesis is to be continued. And if you take these 3 codons, that is UAA, UAG, and UGA, these are the stop codons. That is there are 3 stop codons are there. And whenever a particular during the process of the synthesis of the whether it is the protein, whenever it comes across these 3 any of these 3 codons, then it is a signal for the termination of the protein biosynthesis. And this is the start codon. Try to understand what is stop codon and what are the 3 codons. There is a one start codon is present, and there are 3 different types of the stop codons are there. Any of these is the message or the signal for the termination of the protein biosynthesis. Now we'll come to the last part of this particular protein. There is the genetic code, which is called the Obel hypothesis. Obel has predicted that he has given a particular concept, and what we call it is a Obel hypothesis. And what is Obel hypothesis? The Obel hypothesis discusses about the codon-anticodon interaction. We know that if a particular protein biosynthesis is to be has to happen, and if a particular amino acid is to be incorporated, then what is to be done? There must be an interaction between the codon and anticodon. This is very, very important. And whatever the correct incorporation of the amino acid at a particular place depends upon the how the codon and anticodon are interacting with each other. That's why in case of this particular interaction of codon and anticodon, you can find that the first 2 bases, that is the first nucleotide and the second nucleotide of the codon, and first that is the second and the third that is the nucleotide of the anticodon, they exhibit they strictly follow the Watson and Crick base pairing. Okay, and whatever the third nucleotide is, that is the third nucleotide of the codon and first nucleotide of the anticodon, they do not follow the Watson and Crick. It means that some kind of the flexibilities are there. That is the one of very important point which is there in case of the Obel hypothesis. It means that whatever that particular third position is there, third position of the codon and first position of the anticodon, there is a less stringency. Whereas in case of the first codon and the second codon of the first and the second nucleotide of the codon, there is a very, very strong stringency is present. It means that it will strictly follow the Watson and Crick base. It means that A will only base pair with the U and G will only base pair with the C. But in case of the third position, you can find that it is not going to happen. It is going to exhibit, because we have to accommodate 61 codons for the 21 acid. Then it must be there. That's why I can find that in order to give that particular explanation, they have proposed, Obel has proposed this, OBOL allows several but not all types of base pairing. This is also very important. OBOL allows several but not all types of the base pairing, that we are going to see in the next slide. And now the hypothesis predicted that 2 tRNAs for codon exhibiting the complete degeneracy. It has also predicted that there must be at least 2 tRNAs for those codons which exhibit the complete degeneracy. If there is a complete degeneracy, it means that there must be a minimum of 2 codons, and that's when I find that whatever the number of tRNAs are there, there must be more than 20 tRNAs actually. And in fact, we have got as many as 32 tRNAs are present. And whatever this particular in this particular hypothesis, it believes that for complete degeneracy there must be at least 2 tRNAs for one codon actually. That's also very important. Then the last point is the, if you take the inosine is a derivative, and if it is present at the fifth end of the anticodon, it will base pair with the A, it will base pair with U, or it will base pair with. It means that if the inosine is present at the fifth position, it will make base pairing with the codon at that is the A, or it will base pair with U, or it will also base pair with the C. It means that it has got a lot of possibilities are there for the inosine. And this is the possibilities of the bases. This is the bases in the anticodon. These are the bases in the codon. Now you can find out if the G is present in the anticodon, then it will base pair with the U, or it will base pair with the C. Now you can see that if the G is present in the anticodon, then it will base pair with the U or C. If C is present, it will exclusively base pair with the G, and if A is present, it will exclusively base pair with U. It means that there are only two bases. One is the C and second one is the A. They have the exclusive base pair. C only base pair with the G and A only base pair with U. And apart from that, you can find out whatever the other nucleotides are there. You take G, or you take U, or you take I. You will find that they have ability to base pair with more than one. Actually, G should base pair with the C, but it also base pair with the U. Okay, this is not possible in Watson-Crick base pairing. What's Watson-Crick base pairing says? The Watson-Crick base pairing says that G will base pair with C, and G should not base pair with U. It is not as per the Watson-Crick base pairing. But here, in case of this particular, you can find that there is a less stringency, and because of the exhibition of the less stringency, you can find that G will base pair with the U, or it will also base pair with the C. And in the same way, if U is present, then it will base pair with A, or it will base pair with G. That's why the third position becomes very important. And now you take the I. If the inosine is present, already said that inosine can base pair with A, or inosine can base pair with U, or inosine can base pair with the C. And that's when find that whatever this particular I, U, then G's are there. If these three nucleotides are present in the anticodon, they will make base pair with the that is the first position of the anticodon and the third position of the codon. They will base pair. If there is a third codon, base pair with the anticodon. In this way, we can find out this is the summary of the possibility of the base pairing which occurs at the third nucleotide of the codon and the first nucleotide of the anticodon. If the third nucleotide is either C or U, it will base pair with the G. And if the third nucleotide is G or A, it will base pair with U of the anticodon. That is the first nucleotide of the anticodon. And if there is a C, U, or A in the third as a third nucleotide, then it will base pair with the first nucleotide, that is the I of the anticodon. In this way, this is the summary of this particular thing. I hope you have understood this particular concept. In this video, we have discussed in detail about what exactly mean by central dogma of molecular biology. We have discussed about the definition of the genetic code, what exactly mean by genetic code. We discussed about the features, universal features. They are not just features; they are called the universal features. And that also we have discussed in this particular video. And we have discussed about what is the meaning of partial degeneracy, what is the meaning of the complete degeneracy. Then we have discussed about the OBOL hypothesis. And I hope I have done justification to this particular topic. And thank you very much. And I have referred some of the books which I have referred for this lecture or for this presentation is I have given listed here. My advice is that students should refer the books rather than following the notes, because tertiary and quaternary information is not of any use. You should read the primary information, or at least secondary. Books are secondary information. Research articles are the primary information. You try to read. When you go for PG, then you refer the primary information. But when you are in the UG classes, that is the undergraduate classes, you should refer the secondary sources of information. Try to read good quality books. Don't waste your time in reading substandard books. That is the one important point. And with this, we are going to stop here. Thank you very much for your patience and viewing this video. Thank you.